Thermo Fisher Scientific has been pushing the boundaries of mass spectrometry for 7 decades. Electron transfer dissociation, ETD, is a groundbreaking technique we introduced 20 years ago in our mass spectrometers that's become essential for the characterization of peptides and proteins with post-translational modifications, PTMs. ETD has evolved into a robust fragmentation technology on Orbitrap Tribrid mass spectrometers, providing exemplary results while maintaining ease of use and versatility thanks to pre-built method templates in the method editor. ETD differs from traditional collision-based fragmentation techniques by providing a facile, radical-based fragmentation of the peptide backbone that preserves labile modifications. Preservation of PTMs is crucial in fields like glycosylation analysis, where researchers need to retain modifications to localize the modified amino acids and fully understand glycoprotein structure and function. How does it work in Tribrid mass spectrometers? ETD is an ion-ion reaction that occurs in the high-pressure cell of the dual-cell linear ion trap assembly between a precursor cation and fluoranthene radical anions. ETD results in primarily C and Z-type fragment ions and provides rich, detailed data without fragmenting essential molecular bonds. This makes ETD especially effective for characterizing labile modifications on proteins like those found in glycopeptides and other complex biological samples. ETD functionality is further enhanced by combination with supplemental activation, resulting in higher fragment yield and some collision-based fragment types. This technique combines the softness of ETD with the energy of collision-induced dissociation, generating a more comprehensive analysis of the sample and offering greater insight into the protein structure. When it comes to top-down proteomics, ETD excels. By working directly on intact proteins, ETD provides richer, more informative fragmentation compared to traditional collisional-based techniques, helping scientists uncover details that were previously unattainable with traditional collision-based methods. Whether you're investigating proteoforms or trying to answer difficult analytical questions, ETD gives you the depth and precision needed to precisely characterize biologically relevant molecules that weren't possible to understand before.